Hello, in this video I'm going to talk about water transport across the cell membrane. Water can move across the cell membrane in different ways. It can move via bulk transport, like we talked about with pinocytosis, for example. Water can also diffuse through water channels that are embedded in the cell membrane. These water channels are called aquaporins. Aqua meaning water, porins meaning pore, water holes. Water can also move across the cell membrane via simple diffusion, so it can move right across the phospholipid bilayer. It doesn't do this easily, but it can definitely do it. These latter two forms, diffusion through a water channel and diffusion across the phospholipid bilayer, are collectively referred to as osmosis. So in this video, we're going to concentrate on this process of osmosis. Osmosis is defined as being the diffusion of water through a semi-permeable membrane. So in our case, the cell membrane is the semi-permeable membrane. So like solutes, water diffuses, but we don't call it diffusion, we call it osmosis. Water moves from an area of high water concentration to areas of low water concentration. But if you're like me, you're like, what does that mean to have water concentration? I understand solute concentration, but what do we mean by water concentration, right? Well, it turns out the number of solutes affects water concentration. When solutes are placed in water, it effectively reduces the water concentration. In other words, a high solute amount in the water means there will be a low water concentration. Likewise, a low solute amount inside of water will mean there's a high water concentration. The reason for this has to deal with something called free water concentration gradients, but I'm not going to talk about this in this video. I'll try to set up a link to another video if you're interested in learning more about that. Basically, as I said, water moves from an area of high water concentration to low water concentration, but this is really determined by solutes. The easiest thing to remember is just that water tends to follow the solutes. Water will move towards the high solute concentration area. So let's take a look at this in a pictorial format. Okay, at number one, we have a beaker of water with a semi-permeable membrane down the middle. Okay. Water can cross the semi-permeable membrane, but like a cell membrane, most solutes cannot cross this membrane. At this point, there are no solutes present in the water. At number two, we've added solutes to side B. Remember, the solutes cannot cross over to side A. They're too big, they're too polar, they can't cross, right? But what's going to happen with the water? The water can cross. Now, don't forget, according to osmosis, the rule is that the water will move towards the solutes. So take a second and pause the video and see if you can predict what's going to happen next. If you guessed something like this, you were correct. Water moves from side A towards side B because water moves towards the high solute concentration. Right? So that caused the water level on side B to rise. So that was osmosis in action. Right? Let's use some new terms applied to the situation. The solution on side A is hypotonic. Hypo means lower than normal, and this is referring to the number of solutes. So the hypotonic solution is a solution that has less solutes than a comparison solution. Side B is hypertonic, right? Someone who's hyperactive has lots of energy, right? Hypertonic means lots of solutes. So the hypertonic solution has more solutes than the hypotonic solution. If both sides had the same solute concentration, we would say they were isotonic. The prefix iso means same. So knowing what you know about osmosis, would you say water moves towards the hypotonic or towards the hypertonic solution? Okay. Well, remember, water moves from side A towards side B, so water moves towards the hypertonic solution. Osmosis is a passive process, right? It can occur through a protein in the cell membrane or right across the phospholipid bilayer. But in either case, it's passive and it's not very easily regulated by cells. So if you place a cell in a high or low solute concentration solution, bad things can happen. Cells placed in a hypertonic solution will crenate or shrink because water will leave the cell to follow the solutes, which are highly concentrated outside of the cell. Cells placed in a hypotonic solution will enlarge and possibly burst open, which we call lysis. This occurs because water enters the cell because the water is following the solutes. And if you put a cell in a hypotonic solution, the more solutes are located inside the cell than out now. And water enters the cell and causes the cell to get bigger and bigger and bigger. So let's test and see if you understand the concepts of osmosis. I'm going to have you pause the video and answer these questions, but first let me give you a little information about the pictures at the bottom of the screen. The little yellow plus signs are supposed to be representing a solute that cannot cross the cell membrane. The red circles are representing red blood cells, right? And these red blood cells have been placed inside of a solution. So go ahead and pause the video now, and then when you're ready, replay the video and we'll review the questions. So the first question was, which solution, not cytosol, is hypertonic? So A contains the hypertonic solution because it contains 1, 2, 3, 4 solutes compared to the 2 inside the cell. Over here, there were 2 solutes outside the cell compared to 4 in. So this is the hypertonic solution. Right? Number 2, which cell will eventually lyse? Right? The cell in B will eventually burst or lyse because water is going to enter the cell. Because don't forget, water follows the solutes. Water is going to move from the low solute area to the high solute area. Water always follows that hypertonic solution. So water is going to keep entering the cell, and the cell will get bigger and bigger, and it might eventually burst or lyse. And number 3 says, which cell will lose water? Cell A is going to lose water. And this is because, again, water follows the solutes. There's more solutes outside the cell than in. So the water is going to move from the area of low solute concentration to the area of high solute concentration, leaving the cell, causing the cell to lose water and eventually crenate and shrink. So let's test your understanding of osmosis again. Here we have 3 different scenarios, A, B, and C. In all 3 cases, we have red blood cells, but we have red blood cells in 3 different types of solutions: hypertonic, hypotonic, and isotonic solutions. So go ahead and pause the video and determine which erythrocytes have been placed in which solution type. If you said A was in a hypertonic solution, you were correct. Cells placed in a hypertonic solution tend to shrivel up because they're losing water. Water is moving outside the cell towards the hypertonic solution. In B, we see what a typical red blood cell looks like, kind of like a donut, but not really with a hole in the middle. When red blood cells are in an isotonic solution, they're neither gaining water nor losing water, and they maintain their typical shape. Red blood cells placed in a hypotonic solution, as we see in C, will eventually enlarge and burst open, and this is because water is moving into the cell, because inside the cell has a higher concentration of solutes than outside the cell. If you put a red blood cell in water, that's what we get in C here, because water is hypotonic. Water contains no solutes if it's pure water. So if you put a red blood cell inside of pure water, the red blood cell has stuff inside of it, right? The water will move into the red blood cell towards that hypertonic solution inside the cell.